Hi, Victoria. How are you?
Hey, Katarina. I'm well, thank you. So I'm just arriving to the school, and then I'll get in a space where I can speak
Yep,
better.
perfect. We still have time.
So, yeah, good. How are you doing?
Good, good. Thank you. Everything is good.
Okay, good. Well, I'm just gonna navigate for a minute silently, and I'll be back soon.
Yep. Yeah, I will post a folder with a presentation and so on. So, talk in a minute. Hi, everyone. Today, the quite famous Dr. Feldman is coming here. And I posted the folder, and I will repeat the information later, with a few files, PowerPoint files, because the presentation is big with animations and so on. So I had to kind of split up the PowerPoint in 4 files. So in the title are the slide numbers. So when we move along, you would need to switch to the next PowerPoint file. But I'll repeat this information later on. Thank you. Hi everyone. We will be starting in a few minutes. Dr. Feldman, he's a quite famous neuroscientist who created the field that we will be talking about today. He discovered the brain regions that are responsible for breathing and how they influence different other brain regions. Yeah, he did a lot of pioneering work, and since he's also a medical doctor who sees patients, he also applies this knowledge for health. So, yeah, he's a really very nice and also very prominent neuroscientist. So, I'm really looking forward to this discussion, and we will start soon. And I posted a folder link because the presentation was quite big, so I wouldn't just open, and I put in the file names, the slide numbers. So along the presentation, you would need to click on the next file to follow along, and that way it will work out. So there are like animations in there and so on, which makes the file relatively heavy. Yeah, and in the chat, feel free to check out the paper also. That was like a review that was recently published, and also Dr. Feldman's professor. Oh, thank you, Robert. Sure. Thanks for stopping by, and we will start. Thank you. Yeah, in the meantime, if you have a chance to look up Dr. Feldman, he also wrote books, and there are links where he was interviewed before that I will just share really quick in the chat also. He has a BBC interview that he shared with me that is really good, and also the Huberman Lab podcast. Not sure if you know about that one, where he was interviewed too, which are quite interesting to check out too. Share it in the chat if you want to later on follow up and dive deeper into his work, which is really interesting. Hi Jack, how are you? Can you hear us? So to unmute, there should be a microphone button all the way on the bottom right
Got
corner.
it. Can you
Yep.
hear me now?
Yes.
Okay.
Hey. Perfect. How are you?
Okay. Okay, great. And we're just audio, right? There's no video.
No, there's no video, and I so on top of the room is a link to the Google folder I created for the presentation. But since it's not a screen share, feel free to I don't know if you need to look at the slides while presenting to use your own computer to look at the slides separately from this Clubhouse app, because everyone will access the PowerPoint through these links here. To the link,
Okay, so how do I get them to the right image?
so it's really helpful to say, like when you switch slides, that we are going to the next slide, or maybe the slide number, to mention it. And people are quite used to it here on, like, if they come to this club. So it shouldn't be an issue. The thing was, the file was quite big. So what I did, and I hope that's okay with you, I split the PowerPoint file in like chunks of around 30 slides. So
Okay.
and then I put in the title, which slide numbers are in
So
it.
do you want me to run through the slides, or we're just going to go sort of a Q&A?
Usually a mixture of both works well. So people that don't know your work really well, that they can you know, they get a little bit, but it's really up to you. We can also do, and then usually more in the second half would be more like a discussion question.
Because I didn't really prepare a PowerPoint talk. I thought we were going to have mostly a Q&A, and these were
Okay.
just sort of supplemental. And I could also run through the slides. I mean, whatever you prefer, whatever your listeners prefer.
Um, yeah, I mean, both, as I said, both works really well. Maybe like a broad overview of the brain regions and like
All
would
right, so
maybe
probably the
we
best thing is to let me go through a couple of slides, and then we'll determine whether or not we should continue or go to Q&A.
Okay, perfect. And before
Okay.
we do that, we usually, if it's okay with you, I'll introduce you a little bit to the audience, and then Victoria usually asks like a couple of broader interview questions, and then we go into your research if that's good with you.
Whatever you want. I'm here to do what you feel is
Perfect.
optimal. Sure.
Perfect. So, welcome everyone to Science Society, and of course, a special welcome to you, Jack. We are so grateful that you made time to come here. This is really wonderful, and your work is so interesting. But before we start, just in case somebody doesn't know, Dr. Jack Feldman, he's a professor in neurobiology at the University of California in Los Angeles, and he really started the field of neuroscience and breathing, and discovered the brain regions that are responsible for breathing, and how this breathing patterns and the neurons that elicit basically breathing, how they are interconnected with other brain regions, and how they also kind of um, change behaviors. And he did that with really modern neuroscience techniques such as whole cell patch clamping, recording techniques, to really deep dive into the cellular mechanisms and the synaptic properties and also synaptic interactions. So, yeah, Professor Feldman, he's also a medical doctor, and he really made that, yeah, as I said, and completely changed our understanding of the neural control of breathing, and made groundbreaking discoveries that how respiratory rhythms are generated. So thank you so much for coming again. And Victoria, I'll hand over the microphone to you. Thanks.
All right, thank you so much. And before I begin, I want to say hello, Jack, hello, Katarina, and
Hello.
hi. Can you both hear me okay?
Yes.
Yes.
Okay, fantastic. All right, so yes, welcome, Jack. Katarina and I were especially excited and interested to come upon your research, and then just knowing that you were going to be able to come here is something that we've been looking forward to. So thank you so much.
My
And
pleasure.
yeah, it's great. So we're looking forward to, your discussion, or yeah, I heard you say, however you want to present the information. And before we go into that, maybe we can learn a little bit about you. So I'd like to ask you if you can tell us about everybody has a unique connection to their passion, to what they're interested in. We're here in Science Society, so perhaps you can think back through your life where you first noticed that connection to science in your life, and that could be in your, in your childhood or anywhere along your path, really.
Oh, well, let me see if I can keep this brief. Um, I grew up in Brooklyn, and we were very verbally oriented amongst my friends, and some of them were extremely quick-witted and very funny and smart. And I felt that I didn't want to make my way in the world by just being witty with words because I felt that I really wanted to understand how the world worked, and I wanted to find some things that were sort of independent of whether I was clever or smart. I wanted to understand things, and I wanted to be a physicist. And I went to graduate school to get a PhD in physics, and in particular, I was interested in. I read some philosophy when I was in high school, and I got very interested in the conception of space and time. And I realized that from a philosophical point of view, it was mostly words. And so I wanted to understand space and time from a physical reality. So I wanted to become a physicist studying the physics of space and time, basically cosmology. And I went to graduate school with that intent. And while I was in graduate school, I got interested in the perception of space and time. There was a time in the late 60s where people were exploring various ways of, I might say, altering reality. And I realized that my perception of space and time really was extremely interesting. And I couldn't figure out how it related to the conception of space and time. And I wound up realizing that I really wanted to understand how the brain worked, because that's where the perception lies. And ultimately, our conception is also brain derived. And so I got very interested in neuroscience and read up like crazy. And when I went to my physics department advisor, he got very worried because he felt that the brain was too complicated. And he insisted I try and find something that was pretty simple. And I looked, and I thought breathing would be a behavior which would be not only interesting and important, but relatively simple to solve. I mean, how difficult could it be to imagine how neurons would generate a rhythm? And so I started out on that path. And what I quickly found out was that it was a lot more complicated than I realized. And I spent the early part of both my thesis and postdoctoral studies and academic career sort of upsetting the paradigms that the field was based on, in part because I was not trained as a biologist, so I can ask a lot of questions that biologists were basically taught to ignore. And in asking them, I found out that a lot of the things that we had used as a foundation for understanding how the brain generated breathing movements was fundamentally wrong. And so I started out on a path to try to discover that, and I'm still working on it. We made some fundamental discoveries, which hopefully I can talk about. And now we're very interested not only in the fundamental aspects of it, but also how breathing can influence all sorts of behaviors like emotion and cognition. Hopefully that wasn't too long-winded, Victoria, and we can go on if you want.
Oh, thank you. It's no, certainly not too long-winded. If we had, we were also saying, if we had a whole weekend, I'm sure that you could find lots of things that would be fascinating and really would make everybody want to go deeper into those things as well. It's interesting to hear that that your advisor had helped you to focus your study, and it's interesting, people that you had sort of driving your path was your own curiosity and then the people along your way. So it's a beautiful thing that was sort of that advisor guided you to and then you discovered breathing on your own.
I felt I was very lucky in finding it because one of the great things is if you can find a thing that can occupy your time that you love doing. When people come to me and are interested in working in my lab, I say you can work for a while, but if you find out that it's not really interesting for you, that you don't wake up in the morning thinking about it and go home late at night thinking about it, then it's too tough a problem. But if you're passionate, it's a wonderful life. And I've been really very privileged that I enjoy doing it. I'm curious. I can't wait to think about the problems. I have other things in my life, of course, but it's not like it's drudgery. It's something that just keeps me constantly motivated. And I think anyone listening to this who's thinking about doing science, make sure you choose a problem that you find you care about, because if you don't care about it, you're not going to be able to do it well enough.
Jack, thank you. And that's a really nice example of how these rooms are so useful for people, because we don't always know, things aren't always linear, and we don't always know what we're going to be able to take away from an experience. And so you sharing that, which you noticed about what is a passion that will really speak to you and how to tell, that's valuable information. When we're at a crossroads or wondering where shall I put my time.
Mhm.
And so thank you for sharing that. So then, can you take us along a path of how you've come to this work? I know you brought us up to studying breath, but this particular work that you're sharing today, from where we left off, can you bring us
Um,
up to here?
in order to study how the brain is generating breathing rhythm, we needed to find where the engine was. So, if you have a car and a car moves forward, if you want to understand how the car works, you need to know where the engine is. And that may or may not be obvious depending on what car you're looking at. It could be in the front of the car, it could be in the back of the car, or it could be an electric vehicle, which has no typical engine. It's a very special kind of way of looking at it. And so we looked at breathing. We said, okay, where is the engine for breathing? And people had a general idea, probably going back thousands of years, certainly in Western culture, that it was somewhere in a region of the nervous system we call the brain stem, which is just north of the spinal cord and below the big part of the head, the cortex. And it's a very important part of the entire nervous system because a lot of very vital functions with respect to controlling the body are there. The heart gets regulated there, your vocalization takes place there, your sensory processing for hearing is taking place there. Swallowing, chewing, and of course breathing are all taking place in the brainstem. And but the thing is, the brainstem's a pretty big place, and we wanted to figure out where in the brainstem was happening. And we did an experiment that actually had a history that went back to the early part of sort of the early Renaissance rebirth of neuroscience, where French neuroscientists in the late 18th century were trying to look for where in the body breathing rhythm came from, because that was what they called the noe vital, the note of life was where breathing is coming from, because if you stop breathing, you're not alive. And the experiment they did was they took rabbits and they anesthetized them, and they put uh, uh, blunt spatulas to sort of maybe mash, maybe essentially cut the brain and find out where the vital structures were. And they came to the conclusion that when they cut in the brainstem, they could stop breathing. And so they concluded that the critical structures were in the brainstem, but they weren't able to sort of delineate it further. We were able to reproduce that, but instead of using a blunt spatula, we were able to take the brainstem of newborn rats, which were anesthetized, and we then put these brainstems in a special device that allowed us to take very, very thin slices away from the brainstem. And what we found is that as we removed tissue from the brainstem, there was no change in the breathing rhythm coming from the brainstem. That is, the rhythm itself didn't need the body. The rhythm itself came from the nervous system in the brainstem. But where in the brainstem, we found by basically removing parts, and eventually we came to a very, very limited part of the brainstem where the rhythm was being generated. And then we were able to verify this in intact anesthetized animals, that this critical site was generating breathing rhythm. And it actually had no name in any of the atlases, and we took the prerogative to name it. We had previously named another structure in the brainstem called the bCPG complex. And bCPG is not a famous neuroanatomist. bCPG is an appellation of German wine, where we had a meeting and the wine we were drinking was bCPG wine. And so we named this region after that wine. But that region turned out not to be the place where the rhythm was being generated. The rhythm was being generated just close to that. So we call this region that is generating the breathing rhythm, we called it the preBötzinger complex. And that name is stuck. And that is the site which has now been verified in humans as critical for generating the breathing rhythm. So now we know where the principal engine for breathing is, and it allowed us then to begin to try and dissect that to find out what are the mechanisms, how is the rhythm generated, and how is that rhythm transmitted from the preBötzinger complex. Ultimately, it has to go to muscles that are in the rib cage and the diaphragm, which are going to expand the thorax. So that's when you expand the thorax, the lungs expand, and you have inspiration. And when those muscles relax, the rib cage and diaphragm relax back to their resting position, and you get expiration. So we have to be concerned not only how the rhythm is generated, but how that signal gets out to the muscle and how it's regulated, because you want to make sure that you have enough oxygen for your metabolic needs over a broad range of metabolism, ranging from rest to very high levels of exercise. And you want to make sure that you also exhale the carbon dioxide, which is produced by what's called aerobic metabolism. So you're using oxygen, you're producing CO2. You have to get rid of that CO2 at the rate you're producing it, because if you don't do that, you'll throw all cellular and organ function out of whack if there's too much or too little CO2, because CO2, carbon dioxide, is very critical in regulating the acid-base balance of the blood, and every cell in the body depends very critically on having a proper level of acid base. And too much or too little CO2 will throw that off. So if you have too little CO2, it slows the breathing down, so CO2 can build up. If you have too much CO2, then you will increase the ventilation to bring your CO2 down. So an important question is then, how are oxygen and carbon dioxide influencing this engine for generating breathing so that your breathing pattern is appropriate to regulate both oxygen and carbon dioxide? I hope I wasn't too technical there, but certainly I can elaborate further if you have any more questions.
No,
Before
certainly not. I would just like to say at this point, I'm going to wrap up and let you determine the flow of this discussion. And at some point, guests may put questions in the chat or may even want to come up here and ask you some questions as well. So I'm just going to pass the mic straight back to you and leave it there. So thank you very much.
Okay, so I posted some slides, and maybe I can refer to some of the slides. So in the first set of slides, if you look at I think it's about the fifth slide, it's a picture of a watch face. And I started out, as I said, thinking breathing was going to be a relatively trivial problem. That is, it's simple to think about how neurons could generate a rhythm. And so if you look at a watch, and unfortunately, the thing that's posted on Clubhouse may not be animated, so I'll just have to give a verbal description. So you have a watch, which is a simple oscillator, okay? You have a hand which goes around and around, so that's an oscillation. And you might think, well, it's simple to drive that. All I need is a coil, and as the coil uncoils, it'll move the hand around. But we know that's not what the interior of a mechanical watch looks like. If you if you can't animate this PowerPoint, I would just go online and look at an image of a mechanical watch, and you see all these gears. There's lots of gears. Why do you have all these gears to generate something that's just a simple going round and round? Well, the watch has to be accurate, it has to be precise, it has to be robust in case of shock. You have to be able to change the time if you need to change the time. There are all these things going on, and to fit all those things, particularly in a handheld watch, is pretty complicated. You need lots and lots of gear. So it's not just simply something uncoiling. And it's the same thing with breathing, because breathing is not simply like a bellows, which you just expand and contract. You have to, it has to be robust. You take about between 600 million and a billion breaths in a normal human lifetime. It has to be reliable. In fact, breathing starts before we're born. It starts in the third trimester, and that's essential because you have to be breathing as soon as you are born, and that's not the time to figure out how to do it. The circuits have to get wired up properly, and the way they get wired up properly is during the third trimester, there are what are called fetal breathing movements, where even though there's no air being exchanged, there's movement of the respiratory muscle, there's mechanical actions on the developing lung, which is critical for the lung to develop properly. So that's all happening in utero. And so when you're born, you take that first breath and it's then there for the rest of your life, for up to maybe a billion times. It has to be extremely labile. So, things that are robust don't have to be labile. In other words, for example, if I want something robust, I can imagine a block of concrete just sitting on the ground. It's robust because no matter what I do, it's not going anywhere. But if I want it to be labile, if I want to move it around, well, that's a serious challenge. And the way you could imagine doing it is you put wheels underneath the concrete block. But that's not a trivial solution. Nature had to figure out how to make the system both robust and very labile. And what I mean by very labile is that at rest, you're consuming about 250 milliliters of oxygen per minute. That's about the volume of your fist. That's how much oxygen your body is using every minute. Now, your breathing at rest is sufficient to take in enough air that you can extract 250 milliliters per minute from that. But if you get up and just simply walk down the block, you're suddenly consuming 3 times as much oxygen per minute, 750 milliliters per minute of oxygen, just to walk down the block. Now, the reservoir that you have of oxygen in the blood is not a lot. It's only about 1,000 milliliters, 1 liter of oxygen. So you have to breathe continuously. You don't have a big reservoir to dip into. So now if you go from 250 per minute to 750, if you don't increase the ventilation, you're going to deplete the oxygen in your blood to the point where it won't be able to sustain brain function. You would pass out. So your breathing basically increases the instant you stand up to walk. In fact, it increases just as you're thinking about getting up. And then your ventilation increases very quickly. So that's pretty amazing. And it's very quick, but the brain has to be able to do that. It has to be very long lasting. It also has to fit in with a whole variety of other things which involve the movement of air. The ability to speak, for example, or if you're a non-human mammal, the ability to phonate, to generate sounds. I have to control my breathing. I have constant expiratory airflow over my vocal cords. And we don't notice it, but when someone is talking, you say, oh, gee, they won't stop. But they actually do stop. They stop and take inspiratory efforts during a long speech, or you go to the opera, you listen to an aria, and they seem like they're exhaling continuously over minutes. But if you listen carefully, they're inhaling. So now I have to adjust the behavior to be able to maintain oxygen and get rid of CO2 while I'm doing a completely different behavior, phonation. We have all these reflexes. We have coughing, we have sneezing, we have yawning. All those also being generated by the same muscles. And so the circuits that are involved in generating the normal breathing have to be able to reconfigure themselves for these other behaviors. On top of that, we now know that breathing can have a profound influence on all sorts of emotional and cognitive functions. And we can regulate breathing to have an effect on that. You take a single deep breath, and it's relaxing. We all do that if you're about to participate in some athletic event and you're at the starting line, or trying to hit a baseball, or doing something that's in some sort of match. Beforehand, you take a deep breath and it relaxes you. And we also know that, and this goes back thousands of years, that breathing practice in things like yoga are very powerful in both short-term effects on emotional and cognitive state, but also long-term effects. So breathing practice over a period of time can reduce anxiety, can have positive effects on reducing depression, and that comes directly from breathing. And one of the things we're trying to discover what the mechanisms are that breathing is affecting this. I'm open to more questions. I think well, let me see what I have here in my slide deck.
Yeah.
So
Oh, go
I'm
ahead.
sorry, go ahead.
No,
Please
no.
go ahead.
Well, yeah. I'll just ask a quick question and then please continue. Like, the is there there's a specific neuron type, right? That is necessary for breathing patterns. Is there like can you lose one half of the brain, basically of the spinal cord, and still breathe well enough? Like, do we need both sides, or are we fine using just one hemisphere of the brain in that instance?
Okay. Now, we're not talking about the brain, we're talking about the brain stem.
Yes.
So we're not talking about two hemispheres, and we're not talking about the spinal cord. The engine is in the brain stem. Now, one thing about injury to the brain stem is if you get injury to the spinal cord, depending on where it is, it's usually survivable, but there'll be a loss of function. If you get injury to the cortex, like a stroke, you may, if it's localized, you may lose some function, but once again, they're often very survivable. If you get injury to the brainstem, it often is very serious, and there's very high likelihood of morbidity and mortality because there are so many vital functions which are within the brain stem, and one of which is breathing. And so if you get a severe damage to the brain stem, you can it can totally disrupt your breathing pattern to the point that it's not survivable. Now, we did an experiment in rats. We were able to slowly ablate, that is, remove neurons in the preBötzinger complex. And what we found is that removing up to about half of those neurons, the rats continue to breathe quite well. If you got beyond half, there was a significant deterioration of breathing pattern. And as more and more of those neurons were removed, the breathing pattern deteriorated more and more to the point that it was not survivable. Now, in humans, if you look in Parkinson's disease, at the end stages of Parkinson's disease, individuals have serious problems of breathing during sleep, but they can breathe quite normally during wakefulness. But when they fall asleep, they have serious problems breathing. There is another neurodegenerative disease called multiple system atrophy, where similarly they have they're capable of breathing during wakefulness, but have extraordinary difficulty breathing during sleep. So of course, this is going to disrupt the sleep cycle, which in and of itself will have issues with respect to maintaining body function. In these individuals, when they get to loss, okay, let me backtrack a bit. When you look in the preBötzinger complex of individuals who died at the end stages of Parkinson's disease, and you compare them with individuals who are same age, same weight, but did not die of neurodegenerative disease, but died of other causes, there's a loss of about 60% of the neurons in preBötzinger complex. So at the time of death of Parkinson's disease, there's severe loss of preBötzinger neurons, and that's associated with severe problems of breathing during sleep. In multiple system atrophy, it can get to as high as 90% of those neurons are gone in people who died with multiple system atrophy. And therefore, they have breathing problems. So our best guess is that during healthy life, that there might be some loss of these neurons. They seem to be extremely robust, but you seem to be able to maintain reasonably good breathing function up to loss of about half. If you get beyond half, you start to have breathing problems during sleep. And one of the things that we're interested in, but we have not been able to do the study, is to find out as people die, healthy people die of old age, what the status of their preBötzinger complex is when someone who is 90, 100 years old, when they die, what percentage of preBötzinger neurons are still alive? And is that correlated with their longevity?
Well, thank you. Yeah, I'm trying to ask like some general audience question that I can think of to understand and like the general mechanism. And then does the monitoring of oxygen also happen in the same brain region and then changes the rhythm, and how does it work that the neurons kind of like how does the information pass? Is it calcium signaling? Um, you know, what how does the oxygen monitoring work, and how does it then affect the cells?
Okay,
In this
it's a
system?
good question. Okay, so this principal sensing of oxygen is not in the brain itself. It's in the carotid bodies, which are at the bifurcation of the carotid artery just as it's entering the brain. So the carotid arteries are these very large vessels coming from the heart, going into the brain. And if you feel just under your earlobe, your carotid arteries are located just there, and they're very large vessels. And there's a sort of a bifurcation that splits into two. And at that point, there are special sensors in a structure called the carotid bodies. And these sense oxygen levels. Now, because the brain is the organ in the body which is most sensitive to oxygen deprivation, in order to maintain a healthy body, you got to pay the most attention to oxygen levels in the brain. So having a sensor in a major supply of oxygen to the brain, that is in the carotid arteries, is a good way of knowing what the oxygen level that the brain is experiencing. And the mechanism by which and the Nobel Prize was given to Heymans in 1939 for this discovery, the discovery that the carotid bodies were indeed the source of oxygen sensing that was essential to regulate breathing. The signal from the carotid bodies passes into the brain via what's called the nodose ganglia to the glossopharyngeal nerve, which is the ninth cranial nerve, entering into the brainstem in the region of the solitary tract, which is not the preBötzinger. From there, it gets relayed to the preBötzinger. So the mechanism for sensing of oxygen is, believe it or not, still controversial. There are a variety of ideas about what cellular mechanisms are actually sensing oxygen and how they get transformed into a signal that gets propagated down the carotid sinus nerve and ultimately into the brain. Once it gets into the solitary tract, the signaling there is, you might say, conventional. It's conventional synaptic input. And that input goes from the solitary tract into preBötzinger, and at which point, depending on the amount of activity that's coming in, will regulate both the frequency and amplitude of the breathing rhythm. Carbon dioxide, by contrast, appears to be principally sensed within the brain itself. Here again, there's some controversy, but there's general agreement that the ventral surface of the brainstem, that is the part of the brainstem that would face the front of the body, at its surface, has special receptors for sensing carbon dioxide. And whether they're sensing carbon dioxide directly or pH or acid base is a matter of some controversy. But once again, the signal related to acid base and carbon dioxide is within the brain, which is very important because the brain is extraordinarily sensitive to its acid base status. And so having a sensor in the brain makes a lot of evolutionary sense. And the signal goes from there in a region called the parafacial, that is around the facial nucleus, projecting nearby to the preBötzinger complex, where it's going to regulate both the frequency and depth of breathing. And when the signal gets strong enough, you not only generate inspiratory movements, so at rest, your inspiratory movements are active. That is, your diaphragm contracts, your external intercostal muscles contract. That expands the thorax, get inspiration, and then you relax. Expiration is passive at rest. However, when you have to increase your ventilation significantly, expiration becomes active. That is, you start activating the expiratory muscles, which include the abdominal muscles. When your abdominal muscles contract, that squeezes, pushes the diaphragm up, squeezing the lung. And your internal intercostal muscles, that is the muscles between the rib cage, they pull the rib cage down and in, and that also activates expiratory efforts. So here is a tie between a need for increasing ventilation because of the low oxygen, high CO2, to increasing inspiration through either both frequency and amplitude, but also increasing expiratory effort to allow more ventilation per minute to increase the amount of gas exchange to get more oxygen in and blow off more CO2.
Well, thank you for that explanation. That was really wonderful. And so, to get back kind of how breathing patterns influence emotion, you've shown that that there are various connections to the nucleus coeruleus and amygdala, and then other people showed influence on blood pressure and the heart rhythm. So, since we know that connection, is there any known factor that, let's say, people with asthma, are their emotions influenced by their probably different breathing pattern, and also probably CO2 levels vary during asthma attacks? Are there studies showing that, or is that still something ongoing? Or let's say people with long COVID, maybe are their emotions kind of influenced by that, and maybe then also their mental health state?
This is a complicated problem, and it's very difficult to disentangle all the changes associated, for example, with long COVID or asthma, with how much of that is due to any changes that might be occurring during breathing, or due to some other aspects of it. You know, when someone is having an asthma attack, they have a lot of stress because of the difficulty in breathing. And that stress, irrespective of the changes in breathing pattern, in and of itself can cause changes in emotional and cognitive state. And to distinguish that from the breathing changes that result, I don't know of any research that's been able to surmount that confound. It's been pretty difficult. I think on the other side, um, where one is able to volitionally change their breathing pattern, I think the data is fairly clear that um, changing your breathing pattern can have profound, mostly positive effects on your emotional state. Uh, and it doesn't have to be a clinically diagnosable emotional state. I mean, it could just be that you're a little bit stressed, you're a little bit anxious, and changing your breathing pattern in some structured way seems to be, there's good data that it's very helpful. And I have to say, now, I don't want this to be misunderstood. This is not something that's a recent discovery. I mean, we've known this from Eastern cultures and yoga and meditation for basically thousands of years. The recent renaissance in uh, looking at breathing, I think, is an outgrowth of the recognition of the utility of meditation in dealing with all sorts of fundamental problems that in Western society we've probably been overtreating with pharmacological treatments. And here we have mechanisms that we can uh, take advantage of in our own bodies that are not requiring any drugs and have many fewer side effects and definitely benefit. Now, one might argue, and I have to say, I once felt this way, that the effects of breathing practice or breathing in meditation could be a placebo effect. That is, if you believe it's going to be helpful, then maybe it does become helpful. And know that the placebo effect is very, very powerful. If you believe a drug is going to work or some manipulation is going to work, that can go a long way to getting a positive result, even though the mechanism has nothing to do with the specifics, but has to do with the suggestion that you're going to get an improvement. We have spent a lot of time lately, and after about 3 years, succeeded in being able to get mice to breathe slowly on command. Our idea being is that if we could get mice to breathe slowly on command, basically mimicking certain types of breathing practice, then we could ask the question, does this have an effect on their emotional state? If it doesn't, then you might conclude that maybe breathing practice is placebo. But if it does, then since as far as I can tell, our mice don't believe in placebo effects, that it's a genuine phenomena that we need to study further. So we've now uh, developed a preparation where we can get mice to breathe slowly. These are awake mice. They're put in a chamber and they unstressed, and we can get them to breathe much slower, about 10 times slower than normal. And from all the measures we can make, they seem to be quite content when we do this. And we do it for half an hour a day uh, for 4 weeks. So it's basically like a human doing a breathing practice for a month. And then we've tested the mice. This is a preliminary study. We tested the mice on a standard measure of fear. So psychologists have been studying fear in rodents for a long time, and they've developed very good tests to look at fear in rodents. And when we look at mice put through this protocol compared to mice that we do everything the same, these are control mice, except their breathing doesn't slow down, we find that the mice that went through the breathing practice for 4 weeks were much less fearful. We're in the process of writing this paper up, but I think that I'm comfortable in saying that the results are pretty strong. And what they say are two things. One is that breathing practice or the effect of slow breathing on emotional state is not a placebo effect. It's an effect seen in rodents, and therefore it's not a placebo. And secondly, we now have a way of studying what the mechanisms are. Is it due to olfactory input? Is it due to input from preBötzinger complex? Is it due to this blood gases? Is it due to olfaction, that is, things coming through the nose, because air is flowing in and out of the nose with the respiratory cycle? So we can get down to that and hopefully understand enough that we can see if we can optimize protocols for helping people with dealing with various emotional states.
Well, thank you so much for that answer. And these are amazing results. And I'm really curious to follow the research and also if plasticity changes are happening. So thank you so much. And Nick, you flashed your microphone. Please go ahead.
Hey, Jack. It's Nick Bergraff.
Hey, Nick.
Hey, I had a question, or I guess I was interested to hear your take on it. When you're talking about, the complexity of untangling the relationship between emotion and breathing, um, a lot of that becomes more complex when we try to study it in the humans versus the rodents, because obviously, we know so much about the preBötzinger complex and its interactions in the rodents, and of course, still uncovering much, much more. But what do you think is the pathway to bringing some of that into being able to study it in the human, or is it more beneficial, at least in the short term, like you're doing right now with the slow breathing, to bring some of the human emotion states into the rodent model?
Well, we've been working with Helen Lavretsky, who's a professor of psychiatry at UCLA, who, in treating her patients, uses breathing practice. And she also has a lot of studies using brain imaging. And we are trying to put together a Center for Breathing and Motion where we have a dialogue between those of us looking at mechanisms in non-human animals, particularly rodents, and humans. And so the kinds of experiments you can do in humans are more limited, but you can focus on, for example, take an individual who is diagnosed clinically as anxious. You put them through several weeks of a particular breathing practice, and you compare the imaging and activity in various parts of their brains associated with anxiety, depression, whatever their clinical diagnosis is. And we can see whether activity in those regions are changing. Now, to focus on this, we have a project to map in rodents the ascending projections from preBötzinger through places like locus areus and parabrachial nucleus through into the cortex to see whether we can identify regions that would be good focal points for studying doing human imaging. So I think that that's one way of going about it. And I think until you have some results like that in hand, I think it might be difficult to pursue it further. But and I should say, I should add another thing is that unfortunately, imaging of human brainstem is very difficult. The structures are small, and there are significant movement artifacts with every heartbeat and every breath. and it takes a lot of controls in order to get a reasonable image, and the spatial resolution is not yet sufficient to really disentangle some of the structures. Nick, does that address your question?
Oh, absolutely. And then if I could, just a quick follow-up, you know, especially after, our meeting in Japan, when we talk about some of the more basic mechanisms of rhythm generation, whether it be synchrony or group pacemaker hypotheses and all of these different hypotheses, um, a lot of that is either done in modeling or rodents as well. And do you think bringing that basic rhythm generation type of study into the human is something that is necessary, or do you think that most of the rodent mechanisms are assumed to be, well enough conserved in the human that we should jump to the more complex emotional interactions?
I am loath to stipulate that what we find in rodents is easy to identify an equivalent in humans. For example, when you look at the upper airways, in rodents, the upper airways are fairly straight. In humans, there's basically a right angle. And there are all these functions that are going on in the upper airways that have required a significant amount of re-engineering as humans went from being four-legged creatures, as mammals went from being four-legged creatures, to primates and bipedal and having an airway which goes at a right angle. So there are lots of changes which are going on. And for example, with sleep apnea, you basically don't get sleep apnea in rodents because they don't have airways constructed like ours are constructed. So I think one has to be a little bit careful. I think there are ways to get at what may be happening in humans, but not necessarily by studying patients. I think we've underutilized the study of postmortem tissue. So, for example, you have individuals who have had severe sleep apnea or have had various neurodegenerative diseases. I think we could do a lot more work in trying to understand what the associated changes are in regions that we believe are relate to preBötzinger, parafacial, and whatnot. And I think that could be extremely illuminating about what's going on in humans with respect to the control of breathing and how that might affect other states.
Thank you.
Yeah, thank you. I wanted to follow up a little bit about the anxiety-related study, if that's okay. Did you, because we were talking now to translate this to humans, I don't know if this is ongoing or something you're planning to probably look at in the amygdala, if activity, maybe excitability changes in the slice, and then, but I would assume that translating this to humans, we have to additionally look also into prefrontal cortex area, since fear and anxiety in humans is kind of different, higher versus lower order brain region related emotion. Like there's maybe a bigger difference. So, do you think that makes sense? Or yeah, thank you.
Well, in fact, we are trying to do this. We are proposing to do, or my colleagues are proposing to do, basically whole brain imaging. What we hope to do with the neuroanatomical studies is to try and find regions to focus on to get higher resolution. So, for example, and the thing is, the places that we want to look at first are the places that have direct projections from the brainstem breathing centers to amygdala, through prefrontal cortex and whatnot. Because here again, we want to disentangle what breathing is doing versus the overall change in state. So if someone is anxious, for example, to have a certain brain state, as they become less anxious, regardless of the reason, they're going to have a different brain state. So the question is, if we engage in a breathing practice, what part of that transition in brain state is explicitly due to the breathing practice? And this is part of the challenge of doing the human studies in the absence of a lot of information about the details, for example, the connectivity. And we're hoping that we can get some of that information from our studies in rodents.
And do you, in rodents, do you plan also to look at the glia? Because I would assume that the glia probably, it's more and more, we know more and more information that glia are more and more involved in actual signaling, amplifying signals, and so on. So are you also, and probably measuring also acidity levels and so on. So are you planning to also look at the glia?
I have to be a little bit careful here. I certainly subscribe to the importance of glia. Given the effects that you can see with even just a few breaths on emotional state, I tend to favor that the glia are probably not directly involved at that level. Now, when we talk about a breathing practice, which is sustained over time, involved, and you get real plastic changes in the nervous system, it's certainly reasonable to conjecture that the glia are also playing a role. But for the time being, we have our hands full trying to understand what the basis is in terms of just neural activity.
Yeah, I see. Yeah, that makes sense. But I was thinking that if you make a longer term connection to like healthier ways of living, like to long term health, I guess the glia would be connecting more to the immune system, inflammation states, maybe. I don't know, but maybe I'm going way too far. And I saw Eli, you unmuted, so please go ahead. Thank you.
Yeah, so there are a couple of that raises a couple of related issues connected to long COVID. As I'm sure you know, one of the possible effects of COVID is dysautonomia, and in addition, in long COVID, it's not a 100% marker, but it appears to be a pretty decent marker. Is elevated cortisol, which of course can be a result of stress or anxiety. So first of all, I'm wondering if you think that there could be a connection simply due to kind of, you know, impaired regulation of breathing, before you would necessarily call it dysautonomia, but it's kind of like on the spectrum on the way there, is the first issue, and then there's another one after that.
Um, I'm a little reluctant to really say very much about this because this is really new territory for not just for me, but I think for uh, um, understanding the role of breathing in non-breathing related functions. And then disentangling something like long COVID, which is going to affect uh, a lot of cells, both in the brain and outside the brain, to lay down a particular causal path between effects on breathing inducing some of the changes that you see. I think it's entirely open to speculation. I think I tend to focus on things which I see are attractable experimentally. And to be candid, I'm not sure I see what a uh, clear-cut experiment would be that would disentangle all the confounds.
Yeah, it's definitely a complicated topic. Also complicated, and also involving microglia again. Um, there have been some papers about um, neurodegenerative mechanisms in long COVID or COVID relating to glia or microglia causing damage to dendritic synapses. In other words, causing dendrites to lose synapses. To the extent, and this is of course another open question, but to the extent that a pathway such as that might underlie dysautonomia, do you have any intuitions on whether that kind of damage mechanism, as opposed to losing entire neurons, might be kind of might offer approaches to reestablishing synapses when the neurons are still there?
I, I think, no, I don't have any particular opinion or bias. I think right now it's an open game, and I think we have very little data to uh, distinguish amongst the various possibilities. I wish I could be more informative, but I just don't know.
Well, that is certainly a valid answer, and I think a lot of people would like to know
Yeah,
a lot more.
yeah. I get that. That I get.
Yeah, thank you so much for those questions. Do you think that having all this data from Apple Watches with breathing patterns during sleep and awake and oxygen levels that can be monitored and heart rate levels at the same time, do you think those, I don't know, are those, if you ask Apple, would they give you data sets, and would that be helpful for like a large population type of public health um, approach to see how different breathing patterns long term in people kind of affect health and cognition maybe?
Well, the Apple Watch, the Oura Ring, and whatnot give a lot of very useful information. But about breathing, all they basically give is breathing frequency, which is only one variable which can be important to know. So I think that the information that these devices get right now is too limited. Uh, it's possible that someone could sort through that data and find something interesting, but I'm not so sanguine that would be the case. Um, and to be quite candid, this is way beyond my pay grade. Um, I, uh, I wouldn't even know how to design a study to, to get at that. So lots of people interested in this. There's a lot of people now trying to explore the relationship between breathing and disease and emotion and whatnot. But that, I, and every once in a while you see a paper which has some considerable interest about it. But it's a field which is just, I think, beginning to grow, and it's going to grow quickly. But I'm not really have the I don't really have the expertise on it.
Yeah, I think it's really interesting, and yeah, it's such a easy thing, I guess, to measure and to change. So that's why I think it's maybe easier to create good breathing habits than good eating habits. Maybe that's why people are interested.
Well, there are things like, for example, breathing through your nose rather than your mouth. I think there's good data that nasal breathing, by and large, is better than mouth breathing. Um, so I think that that's a step forward. It's not that you cannot mouth breathe when you certainly have to generate high degrees of ventilation. Like when you're doing serious exercise, you need to get air in through your mouth. But by and large, nasal breathing seems to be better along various metrics. And so, so I think there are some benefits which are coming from these studies, but we have a long way to go before we fully understand that.
Yeah, and another thing is, did you check if it has, like, if you change the breathing patterns of rodents during pregnancy, if that changes maybe also the anxiety levels of the offspring. Is that something you may be interested in or have some preliminary data on? That would be interesting.
Well, it would, this would take a significant part of the NIH budget to do all the experiments that I think are important to sort of sort out. We're very focused on a very limited set of it, but of course, if the pregnant mother is pregnant dam is um, less anxious, it would be interesting to know what the outcome was in terms of their offspring, not just whether they breathe slower, but is there any physiological parameters changed? Are they healthier? Are they calmer? Are they more or less uh, cognitively able? I mean, there's lots of things that could be done.
Yeah, please, Susan, go ahead. Thank you, and thank you for that answer.
Hi, I was wondering if you're familiar with box breathing and if you could tell us, if you are familiar, why you think it's so effective.
Well, I would say my breathing practice is box breathing. Uh, My pal Andrew Huberman tells me that that Navy SEALs use this when they need to calm down acutely, and I would imagine that they face more stressful situations than I do day to day. Um, and it's a very simple practice, and I encourage people who want to explore breathing practice to start with box breathing because it's very, very simple. The likelihood of having a negative side effect probably is probably very, very small. And there are a variety of free apps you can get with your smartphone that make it easy to generate a practice. And so, for those of you not aware, box breathing basically is inhale, hold, exhale, hold. So you can imagine sort of going around the box. And uh, typically I do 5 seconds, 5 seconds, 5 seconds, 5 seconds. So I'm breathing now from a normal breathing rate of between 12 and 15 per minute. I'm breathing now 3 per minute. And I find that when I do that, it's very restorative. Either if I just do it for 5 minutes or 10 minutes, it's very restorative. But, I'm an underpowered experiment. I'm just giving you my anecdotal observation. And I would encourage people who have interested in trying a breathing practice to just get a breathing app and try it. And try it for 1 minute, then 2 minutes, then 3 minutes, then 4 minutes, and if you feel comfortable. And if 5, 5, 5, 5 doesn't work, try 4, 4, 4, 4, or 3, 3, 3, 3, and see whether or not you notice any changes.
I also,
Did that answer your question?
yeah, I mean, it does because a lot of breathing exercises either don't work or I find them not helpful, but this seems to work really quickly. And also, I know a lot of VAs try to incorporate this because it minimizes intrusive flashbacks. And I guess because it is so easy, so I thought that wasn't really helpful. But thank you for also, confirming what I thought. Thanks.
No, I mean, there are lots of breathing practices. There are so many which slow down breathing, like box breathing. There are some which speed up box breathing. There are some which, when you do them, can be quite taxing. And well, I liken it to when somebody asks me, I don't exercise, what exercise should I do? And my response is, do something easy to start. Get up and walk for 5 minutes, then walk for 10 minutes, then walk a mile. And when you get comfortable walking, you can begin to think about other things. And uh, you then have to make a decision about what you want to optimize with your exercise. You're interested in general health, you want strength, you want endurance, and then you might pick a particular exercise. And I think it's the same thing with breathing practice, that something like box breathing is sort of like easy and you could do it, and then you can go from that and try out some other breathing exercises which may be a little bit more challenging or may better fit your own lifestyle or temperament, and find something that works for you. And if it doesn't work for you, that's okay. I'm not being judgmental about it, but I think you need to sort of get up off the couch and do something in order to see whether it works.
Yeah, thank you so much for the question. That was a really great question. Kyle, I know you had a question or two. Please go ahead.
Yes, thank you very much. It's amazing that you're doing this work, and I heard about um, kind of your I guess adventure to find the engine. Um, I'm curious about whether or not you measured the body temperature of the animals that you were studying, and also if you noticed a holding of the breath at some points in time.
Okay, so a lot of our advances actually came not from whole animal studies, but what are called in vitro studies. So we would take slices of the brainstem of neonatal rats that were sacrificed, and these slices contain the preBötzinger complex. So the whole animal is not there, but it's a little bit like trying to study the engine of a car. Is it easy to study it when the car is going down the highway at 60 miles an hour, or to take the engine out and put it on a table and study it outside the car? And that's what we're doing with these slices. And then once we understand the basics of the engine, we then look in whole animals, which is basically putting the engine back in the car and studying the car as it's moving down the highway. So a lot of our advancements were from these slices, and then we did experiments to verify that they were occurring in intact animals, that we verified them. Um, we in an intact animal, you see all sorts of breathing patterns, and as best we can tell, they all originate from the preBötzinger complex. So you have the normal breath, you have sighs, you have gasps, you have coughs. They all seem to originate with preBötzinger.
That's interesting. Thank you for that question, Kyle. And the question that comes up is that would probably be interesting for everyone. Like, how does this brain region switch from just automatic breathing to this very mindful type of breathing? And how do you think this mindful type of breathing then changes the automatic or habit type of or unconscious, subconscious breathing?
So, okay, so the breathing is an automatic function that you don't need your cortex to breathe. We know that humans are animals that have had their that are quote brain dead, whereas there's no cortical function, still continue to breathe. This causes some ethical problems in the hospital with individuals who have no cortical activity continue to breathe. And so, how do you proceed with patients like that? Uh, However, when the cortex is working, the cortex can directly control activity in preBötzinger complex, as best we can tell. So when you slow breathe, it's being mediated by cortical inputs into preBötzinger. Now, Victoria, I'm sorry, I lost the thread of your rest of your question.
Oh yeah, so yeah, you asked the part of how basically this would intervene would be through cortical influencing the neurons that are responsible for the breathing patterns. And how does this habit of this, let's say the box breathing that was mentioned, does it then influence longer term the regular breathing pattern that is kind of automatic? Oh, that is automatic. Does it change it long term if you do that once a day? And then how does this work? Does this change?
I don't have the data on that, so I don't know whether the breathing pattern changes significantly or there are changes in blood gases that are significant. My guess is my general understanding is if it changes, it's very small, and uh, but I don't really have any direct data to refer to.
I have read a bit about that with respect to blood pressure lowering in connection to regimented breathing patterns, that after 2 weeks, and that there's a measurable level of drop in blood pressure. And that was, I'm sorry, I don't have a link right now, but it was confirmed. It was,
Uh,
I don't know, it was American Heart Association, or is that what you mean,
No,
Katarina?
well, no.
Like
Okay, let's
physiological
distinguish.
change?
Well, let's distinguish. The question related to whether there was a change in breathing pattern or blood
I see,
gases.
that's stuck.
Okay,
I
okay.
see, I see.
Now,
Breathing pattern, right.
in
Okay.
terms of autonomic function, heart rate is regulated by autonomic output. This is the part of the nervous system, the sympathetic and parasympathetic nervous systems. If you decrease sympathetic activity or increase parasympathetic activity, you're likely to both slow the heart rate and lower blood pressure. And it's been established probably thousands of years that meditation and breathing practice does lower blood pressure. So that's well established. I was addressing the issue of whether your breathing pattern changes per se, and I don't have any data on that. Or I'm not aware of any data on that.
So, since you have the rat model in progress, I'm wondering if you've thought about doing RNA-seq to investigate the effects on metabolic pathways.
To be candid, we've thought of 1,000 man-years of experiments. And the problem that we're facing is choosing which ones are the most important ones. So I I don't know. The answer is yes, we've thought about it. The question is, we haven't really decided where we would put that experiment on the priority list. And my feeling is that that is an experiment that may be of more interest to people interested in metabolism than people in my neck of the woods. And so there's a lot. Once this model gets published, then hopefully lots of people will begin to study all sorts of angles, including the notion of what's happening in metabolic pathways.
So, I mean, the nice thing about RNA-seq is that it doesn't presume much to start with, other than the cells that you're sampling, at least if it's done properly. And so, I mean, the metabolic stuff is a subset of that I've seen used in various papers, looking into various conditions, including the effects of COVID, but. Of course, it goes beyond metabolic pathways, and I mean, the nice thing is that you can spot signals that you would have never thought to look for.
Um, I'm in total agreement with you, uh, but we haven't decided how high a priority that is, because when people have done RNA-seq under just normal conditions, very little has risen to the surface of things that we were not otherwise aware of. So we have a lot of information that has not really driven our understanding. And what we're looking for is for experiments that are going to drive our understanding further. And sure, I would love to have more data on RNA-seq, but given the time and effort that goes into doing those experiments properly, It may be that we could do more important things looking at other metrics.
Thanks.
I had kind of a question about that, Jack. This is Nick again. Someone had mentioned previously, looking at some of the larger data metrics or big data type of metrics from wearables, or whether it be at home spirometry or anything like that. And you had mentioned about coming up with experiments would kind of just take up the entirety of the NIH budget for just a few experiments. Do you think like trying to understand that side of the data being, at home big data type of sets and wearables, regardless of the quality of that data, but there's certainly an enormous amount of data that is there. Is that something you think that will ever be able to be utilized through like an NIH type of funding, or is it going to take a push through a private funding such as, like the Allen, or kind of like how Microsoft took up some of the SIDS research?
Nick, this is way above my pay grade, and I almost feel that almost anything I say, I'll be criticized for things I didn't mean to be I don't want to be criticized for. So I think I'm going to
Yeah,
stay
definitely don't
outside.
do that.
I think I'm going to stay outside that issue of trying to prioritize what should be done and what should not be done. I think
Sure, sure.
big data has proven to be very powerful, but I think until someone comes up with an observation from big data that was surprising, it's a little hard to advocate, well, let's do it because maybe we'll discover something. To do an experiment like this properly, I don't think the wearables give you enough information. And I think you'd have to design in a way that you knew enough about the individuals and their health history to be able to really do like a principal component analysis to try and find out what's really correlated to what. I would embrace someone doing it, and I wouldn't object to it doing it unless they took money from my research. Then I would object.
Perfect. Thank you. I have to take off here pretty soon, so I just wanted to say thanks for coming
Well,
by.
thanks for listening in, Nick.
Yeah.
Thank you. Thank you for your question, Nick. It was remarkably like Katarina's that she asked a bit ago. And thank you, Jack, for answering that so thoroughly. It's such a great question. You mentioned anecdotal observation, and when you were talking with Suzanne about the box breathing, when my kids were little, I noticed that when they were engaged in what we would now call mindful, when they were practicing mindfulness just by default as of being kids who engage in their tasks, that their breathing slowed, that it was something that you could see and even hear, that they were breathing more deeply, there was more space between each breath, and it was very evenly paced. And now I work in arts and science education, and I notice a similar thing with my students, that when they're engaged in art activity that's making them, what I would observe, feel peaceful and really mindfully engage their whole being, the exact same thing happens with their breath.
Well,
And yeah, go
well,
ahead.
one thing we have not talked about. So I've talked about the effect of breathing on emotion. There's another aspect of this, which is the effect of emotion on breathing. And they're they're not two sides of the same phenomena. The effects of emotion on breathing are a whole other set of issues that may involve similar structures or different structures. I can let me do you, let me talk a little bit about locked-in syndrome. Locked-in syndrome is the result of a major stroke in the basilar part of the pons and part of the brain stem. What this does, it cuts off all the signals coming from the cortex to most of the motor neurons of the body. So these individuals are not able to move any muscle except their eyes, because the motor neurons controlling their eyes are not cut off from the cortex, because they're rostral on the other side of the lesion, and they maintain their breathing because all the structures critical for the automatic breathing are on the other side of the lesion and don't require these descending inputs. So these people are motionless except for breathing, but they can move their eyes, typically either blink or move their eyes left and right, and they are normal intelligence. There's a great book called The Diving Bell and the Butterfly, written by a man who had major stroke, had locked-in syndrome, and he basically blinked the book to his caretaker. And it's amazing testimony to the human spirit that he was able to be very lucid in describing what his life was like. I had colleagues who were studying a patient who had locked-in syndrome. The patient's breathing pattern was very, very regular, very robotic. They asked the patient to hold his breath. The patient couldn't hold his breath. They asked the patient to breathe faster. The patient could not breathe faster. But if they gave the patient a little bit of carbon dioxide to breathe, which would normally stimulate breathing, it stimulated breathing. So the chemical regulation of breathing remained intact, and the person was able to maintain healthy levels of oxygen and carbon dioxide. And there was nothing that they could do to get the patient to change their breathing pattern. Then all of a sudden, the patient's breathing pattern changed. And they said to the patient, "Your breathing pattern changed. What happened?" And the patient said, by blinking, said, "You told a joke and I laughed." And it turns out whenever they told a joke that the patient found funny, the breathing pattern changed. So what this says is that there's a pathway that affects breathing that's emotional, that is not affected by this lesion, which affects the volitional part of breathing. So there's a whole pathway devoted to emotional breathing, which includes things like laughing, crying, emotional sighing. And so there's this whole descending system coming from cortex and amygdala and whatnot that goes down and can affect breathing patterns. So when you say that your children their breathing pattern changes, it's probably because their emotional state is something that involves being focused, being calmer, and that in of itself is now sending signals that are affecting the breathing pattern that comes down through this emotional pathway as opposed to the volitional pathway.
Thank you. Yeah, go ahead, Katarina. I was just going to say that's really deep and beautiful. And I'm thinking when we hear a joke, I can think of times when I'm really tense and a friend will tell a joke to make me relax or help me relax. And I laugh. And just as you're saying, then my emotion changes, then my breathing changes, then I feel
So there's a
relaxed.
loop there. So your breathing changes and that goes back, and now it's going to change your emotional state, irrespective of any direct effect of the joke on your emotional state. Katerina, you had a comment?
Oh no, I wanted to just say the same, that this is really so interesting, and yeah, thank you for the question and also for that answer. Um, and I also wanted to mention that we've been talking, like you've been talking for an hour and a half, so I wanted to ask if you wanted to maybe switch to something else in your, and get rid of us. So yeah, I just wanted to give you a
Well, no, I
chance
don't want to keep your
to.
audience, your clubhouse captive. Maybe what we can do is that if there's interest in continuing the conversation, we can schedule something for some time in the future to follow issues that may arise from this discussion. And so now may be a good time just to close out. If you have any final questions, I'm happy to answer them.
I think that's wonderful. And thank you for that suggestion, because I would have asked you, maybe you could come back in the new year sometime and we can continue this discussion, because it's such an interesting topic, and it's like the example of a, a research you do, and then it opens up a whole new field and a whole new thousands of projects ideas that you could do. So it's like really good port wine. I'm from Portugal. The research. So thank you for that, and thank you for taking the time. This was really wonderful, and we got so many insights from it. And everyone, thanks for asking questions, suggesting questions to me, and really appreciate that. And it makes, I think, the discussion so much more interesting, if not just one person asks, but also other people participate, because then many more topics arise that I wouldn't think of. So thank you so much, Jack, and enjoy the
Well,
holidays.
thank you very much. I think it's a privilege. I think what you're doing here with this type of discussion is very, very important, and it's seriously undervalued, but it's extremely valuable. And for me, it's a privilege to have been invited to participate. So, best wishes to everyone for the holidays, and hopefully we can continue this discussion sometime in the future. I'm going to sign off now.
Yes.
Um,
Thank you so
Thank you.
much.
Bye now.
Thank you.
Thanks,
Thank
Jack.
you.
Bye,
Bye now.
everyone. And
Thank you.
yeah, thank
Thank
you.
you.
That's one. Thank you for this wonderful room. And keep breathing, everyone. just do the box breathing. It can't hurt you. So just do it, and and yeah, enjoy the rest of the evening, morning, or day, wherever you are right now. And if you like discussions like this, we have tomorrow another room with Dr. Cohen. He will talk about autonomous microrobots with artificial brains in them. I think this is the first paper that came out with having done this quite successfully. He will be here tomorrow at 6 p.m. EST. And then we'll have more rooms next week. So check out the club and the calendar. And yeah, thank you, everyone. And I'm closing the room in 3, 2, 1. Bye, everyone. Thank you so much.